So thank you everyone for coming. My talk will be titled Adaptation Genomic in Island, the role of gene flow and structural variants. And my advisors are Carles and Inés. So I would like to start with this citation from Meyer. That's really nice and expresses the importance of island. That is, an island may demonstrate certain biological phenomena almost with the clarity of a test tube experiment. So why do we say that islands are a perfect study system for evolutionary processes? Because they have a limited area and very defined boundaries. Generally, their geological age is well known, and they can be considered as independent replicates of these processes. So based on this, my PhD will focus on two different parts. The first will be an interspecific analysis in which I will try to assess the mechanism of convergence in an island radiation. Well, actually, the first chapter is a methodological one, biased assessment of ancestry in a mixed population using structure. And the second will focus on the role of local adaptation and gene flow in a leaf litter frog species in Cuban. The second part is an interspecific approach in which I will assess the molecular mechanism responsible for phenotypic polymorphism in an island quail. And in the third chapter, I will try to figure out if there is an inversion that could be responsible for these different phenotypes. And in the fourth chapter, I will study the colonization and dispersal across Macaronesian island. So we start with the first part. So convergence is subject to the same, happens when some species or population are subject to the same selective forces. But is this convergence driven just by independent mutation, or gene flow and hybridization play a role in facilitating this convergence? There are, for example, in cichlid from the African lakes, it seems that the hybridization of some ancestral species facilitated this really rapid and explosive reductive radiation. And there are increasing evidences of the importance of gene flow and introgression in a high number of species. For example, in Darwin finches, in Heliconium butterfly, or in sunflower from the genus Eleutherodactylus. But before trying to figure out if there is which role play both factor in an adaptive radiation, I will start with a methodological chapter. I started this project during my master, and I wanted to assess the power of analysis and accuracy of a software that is commonly used to detect introgression and hybridization, which is called structure. Well, you will probably know this kind of bar plots. And structure is widely used because it allows for each individual to estimate the proportion that comes from each ancestral population. But there were some studies that suggested that the accuracy could be not that good when there are many generations of introgression, of hybridization. And so the main question of the chapter was what happens when we don't have a proper reference sample to add in the analysis, and if we are underestimating the real degree of introgression. So to do it, well, I will not enter into details, but I realized a high number of simulations from one population into the other, 10 generations among these two populations. And you change a lot of factors, among them the level of genetic differentiation and the hybridization rate. So yeah, I will show you one significant result from a common situation, that is with an FST of 0.1 and 30 marker with 10 alleles. So in this graph, you see along this axis the real proportion of genome that comes from the ancestral, from the original population. And along this one, the same value, but estimated by the program. So if the estimates were perfect, were accurate, all the points should lie on the diagonal. But as you see, in both situations, most points lie above this line. And so it indicates that we could be underestimating the real degree of integration. However, these results tend to improve when we add references. And yeah. But as I wanted to study the role of gene flow, I want to study the role of gene flow in the past, I will use genomic approaches from this point on. So I will introduce you my model study for the first part, that are Eleutherodactylus frogs. It is a genus that evolved into more than 150 species in the Caribbean islands. And these species are adapted to a high number of microhabitats, such as there are some arboriculous ones, caverniculous, leaf litter ones, and so on. A study published last year from one member of our group showed that it is a good example of parallel evolution. It means that the same ecomorphs evolved in different islands independently. So, for example, if we focus on the bromeliculus, that are the light green ones, we can see that there are some in almost all islands. And moreover, in Hispaniola, they appeared independently at least 3 times, here, and here. However, due to some logistical problem, in my PhD, I will focus only on leaf litter species from Cuba, that are those represented in orange in this tree. And I want to figure out which role play local adaptation and gene flow in leaf litter frog species in Cuba. Cuba has a very interesting geological history, as it was separated into 3 parts until 8 million years ago. And if you see in this graph, 8 million years ago, the 3 main clades had already diverged. Even in more recent geological times, we can see that the 3 main areas, the western, central, and eastern, were separated. Well, this color indicates that in the light gray one, that they were inundated along some periods, and therefore they were not a suitable habitat for frogs. Moreover, these areas correspond nowadays to highland regions, here, and here. And in this highland region, we can find the suitable habitat for leaf litter frogs, that are this kind of forest. And this frog lives in this habitat in the lower part of the forest. And the main feature of these frogs is that they are really tiny. I mean, the biggest one is like 3 centimeters long. And so you can see that the dispersability is can be quite limited. Yeah. These ecotypes, well, we have different species for this ecotype, morphotype. 3 only from the western part, 2 from the central, 5 from the eastern part, and 2 species are present in more than one region. So based on this, I want to find out if the same genomic region are involved in the adaptation to the leaf litter microhabitat. And if it is so, I want to figure out if the gene flow could facilitate this adaptation, especially in sympatric species. So a collaborator of us will provide us some sample from Cuba, 10 leaf litter species and some outgroups. We will sequence the whole genome of these frogs. Well, actually, I have already assembled genome from one species of the genus that will facilitate my work. And then I will compare between different habitats and within the same to find out which region could be under selection. And so, now I will pass to the second part. And in this case, the study system is common quail. A common quail is widely distributed along all the Paleoarctic region. Well, it is a migratory bird, and it lives mainly in grasslands. Along its distributional range, we can find different phenotypes. Two features that change along the distribution are the male throat coloration, that you can see can be dark in some individuals, fair in other ones, but well, we can find some intermediate ones as well. And another different phenotype is the wing shape. They can be more rounded until pointed wings. And it seems that it is related with flight efficiency, and therefore it could influence the migratory potential of these birds. We realized some preliminary genotyping by sequences of about 100 samples of these quails around this part of the distribution. And we found that in this PCR you can see that there are 3 different genetic clusters here. And if we focus now on the Macaronesian island, these two, we see that the cluster C, the black one, is present only in Azores, while in Madeira we just find cluster B, and in the Canary Island, both cluster A and B coexist. And moreover, they coexist in the same island. Then we compare this with the phenotypic traits of these quails, and we see that for these two clusters, for example, B and C, most individuals have more rounded wings, and therefore like a lower flight efficiency. And if we focus on the coloration, most individuals are dark for these two clusters. For example, if we focus on the Canary Island, we try to compare cluster A and B to see where could be located this strong genetic differentiation, and we found this pattern. So this is the level of genetic differentiation among the two clusters, between the two clusters. And along this axis, you can see in different color the different chromosomes of the quails. And in this region of chromosome 1, that encompasses like most of the chromosome, you can see that the differentiation is very high. And we think that this pattern of differentiation could correspond to an inversion. As you know, an inversion takes place when a region of the chromosome changes its orientation, and there is no loss of genetic information, but the relative position of the different alleles change. Inversion were first discovered in Drosophila, but now it seems that they are present in a very high number of taxa. And inversion can play an important role in the adaptation, in local adaptation. Why? Because they suppress recombination. When an inversion is formed, the gametes that are originated among one inverted and non-inverted chromosome can be just like the ancestral one. There can be no recombination among them. Otherwise, there will be a loss of genetic information. And this can be useful in local adaptation. For example, if we consider a population in which we have different alleles that are recombined in different ways, like in this case, if an inversion takes place, like here, and this region contains genes that are co-adapted to a specific habitat, it prevents them to be separated, and therefore it can originate something that they call a supergene, that it is like a group of genes that are always inherited together, like if they were a single Mendelian unit. And so if they give an advantage to the individual that carry them, they will increase their proportion in a population. There are many interesting examples of the role that inversion plays in local adaptation. One of them, for example, are these genus of butterfly, Heliconius, where the different mimetic wing forms are due to the presence of different inversion. And this case, it's even more amazing, is from rainbow trout. Well, they are the same species, but different ecotypes that change in their migratory behavior. And it seems that a double inversion, that's represented here, is responsible for these really huge differences. And based on this, in my third chapter, I will test if there is really an inversion in these quails. And the first approach to test the presence of this inversion is through fluorescence in situ hybridization. We think that the inversion is located on this chromosome, and therefore we design two different probes, one outside the inversion, probe 1, and the other one inside the inversion, well, the region of the inversion. So in no inverted chromosome, we would find that the probe is located here, while if there is an inversion, this probe would be quite closer to probe 1. These analyses are carried out in collaboration with a group in the Universidad Autónoma de Barcelona. And well, here you can see an example. I don't know if it is very clear. We are still optimizing the technique. There have been some problem to get it working. And in this case, it seemed that it could be an heterozygous individual. Why? Because there is one signal for probe 1, this one, but there are two signals here and here for the second probe that could correspond to inverted and a non-inverted chromosome. However, we are still working on it and try to figure out if this is the good hypothesis. So this approach will be complemented with whole genome sequencing, both with PacBio. That's very useful because it gives long reads, but it has a high error rate, and therefore it will combine with sequences with Illumina that provides short reads, but with a very high accuracy. And therefore, the combination of both will provide us with really useful information about the inversion. So far, I have just some results from Illumina that I'm going to show you. Well, it's the same graph as the one we saw before. And we confirm that there is this really big region, very differentiation among individuals that carry the inversion and those that don't. And it is interesting that other region appears that could be compatible with other kind of chromosomal rearrangement. But now if you focus on this region, it has a kind of U shape, like this. And this is suggestive of some kind of recombination. How can it happen? When the two, well, this is a very large inversion. And therefore, if we have a chromosome that is inverted and one that is not, they can form a kind of a loop, like this one. And recombination can occur in the central part here, but not in the breakpoint, as in this region, the two chromosomes can't pair. So we have some other analysis that suggests the same pattern of possible potential recombination, but we are still working on it. And the last chapter is about the colonization of Macaronesian island. Well, as you know, they include Azores, Madeira, Canary, and Cape Verde. In Azores, there is a subspecies of quail, common quail, that is considered to be resident. And as I showed you before, in Canary Island, different morphotypes and phenotypes coexist. But if many birds have this kind of wing, more rounded one, we assume that they have a limited dispersal potential. And therefore, we asked how they could colonize the different islands. And we want to find out as well if the inversion could evolve in island or not. To do it, we will combine an approach of stable isotopes that will be carried out here to figure out if the individuals are migratory or resident. And we will increase the sample of genotype by sequencing to all the islands to test how many lineages there are. And so, well, that's all. This is a tentative timeline of my PhD. My fellowship started at the end of 2018, and it will finish by the end of 2022. I almost finished chapter 1, and I have been working last year on chapter 3, the one of the detection of the inversion. And I've been trying to find samples to carry out the second chapter, the one about convergence in frogs. Last year, I realized a very short stay in the Universidad Autónoma de Barcelona to learn something about the immunofluorescence. And during this year, I will probably go to Uppsala with Matthew Webster to learn more about bioinformatic tools. And yeah, I want to thank my group here and all our collaborators. And just out of curiosity, quail was one of the third birds to be eligible to be the bird of the year from SEO from this year. And well, that's all.